بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. Today lecture is about introduction to medical mycology. So the objective and the aims of this lecture is to understand the basic structure of the fungi, also to understand the general classifications of fungi according to their characteristics. And by the end of this lecture, also you should study the basic fungal pathogenesis, and also to know the modes of fungal transmission, the concept of fungal virulence, and the most common virulence factors of the fungi. Moreover, also to understand the basic of lab diagnosis of fungal diseases, so how to lab diagnose these fungal diseases, and to learn about the basic concept of antifungal agents اللي هي medication we use to treat fungal infections. طيب. Let's go about the medical terminology اللي عندنا هنا اللي هي mycology. What is the meaning of mycology? Mycology means ology, as you know, is science. Myco means fungi. So mycology كلمة نفسها معناتها science of fungi. But what is the meaning of mycosis? Mycosis means any disease induced by a fungus. يعني الأمراض اللي بتسببها الفنجس. Okay? But fungi are eukaryotic cells which lack chlorophyll. ما عندها الكلوروفيل. So they cannot generate energy through photosynthesis. يعني ما هي زي البلانتس ما عندها الكلوروفيل. فبالتالي cannot generate their energy through photosynthesis. So they and also they require aerobic environment to live and grow. They are gram-positive, most of them gram-positive, and thousands of unknown species, but only about 100 of them are pathogenic. Okay, يعني عندنا thousands من الفنجاي, but only وجدوا إنه only 100 of them can cause disease in man and animals. And also many others can cause disease in plants. يعني هي مهمة في plants and also in human and animals because they can cause diseases in them. Some of them are dimorphic. What is the meaning of dimorphic? يعني form different structure at different temperature. يعني فرضا ممكن تنمو في درجة حرارة 22 وتكون شكلها dimorphic. يعني كيف شكلها dimorphic؟ يعني hyphae for example في درجة حرارة 22 whereas إذا they grow in different temperature like 37 or 35 degrees هيكون شكلها spherical اللي هو yeast cells. Some exist as molds in saprophytic environment and as yeast in host tissue. برضو شكلها بيتغير according to its environment. Okay? All fungi lead a heterotrophic existence as saprobes. Okay, يعني organisms lives on dead or decaying matter. يعني بيعيشوا بعض الفنجاي على ال dead matter or dead body. Okay, احنا بنسميها saprobes. بعضها بيعيش as symbionts. Symbionts. Meaning of symbionts يعني live together with another living animals or humans, and in which the association is of mutual advantage. يعني الاثنين بيكونوا بياخدوا يعني ما بتسبب له الضرر، لكن الاثنين بيكونوا ايش عندهم mutual advantage، يعني الاثنين بيستفيدوا من بعض، سواء الفنجس او البادي اللي هي عايشة عليه، الأورجانيزم اللي هي عايشة عليه. طيب ممكن برضو تكون they exist as commensals. يعني إيش؟ يعني organisms living in close relationship in which one benefits from the relationship and the other neither benefits nor is harmed. يعني بتعيش على another organism والأورجانيزم الثاني يا إما ما بيستفيد منها يعني لا بيستفيد منها ولا هو متضرر منها. أوكي، فبتتسمى إيش؟ commensals. Okay, so they can exist as saprobes, sympoints, commensals, and also they can exist as parasites. Parasites هنا هتسبب إيش؟ هتكون pathogenic. They can cause disease at the organism they live in. Okay, هي بتكون ضارة. The characteristic features of the fungi. إيش خصائص الفنجاي؟ أول حاجة إنها ubiquitous. يعني إيش ubiquitous؟ يعني we can find it wherever possible. يعني anywhere we can find it. Okay? Heterotrophic organisms يعني cannot produce its own food. Okay? cells زي ما قلنا eukaryotic, multicellular, filamentous mostly أكثرها بتكون multicellular, with some they are what unicellular وحيدة الخلية، وبعضها بتكون multicellular، وطبعا بتكون aerobic يعني they need oxygen for living. To live they need oxygen. Okay, they are strictly aerobe or facultative anaerobes. Okay. Let's see the structure of the fungi, and certain fungal structure characteristics serve as virulence also. And some of the antifungals, antibiotics, can act on this structure to stop the growth of the fungi. طيب تتكون من إيش؟ بتتكون من cell wall زي ما انتم شايفين هنا اللون الأزرق الغامق surrounding the cell membrane, composed mostly بيتكون أكتر حاجة من الكاربس، فيه له some proteins. أوكي، بيعتبر potent antigens to human immune system. أوكي، يعني بيحفز الاميونيتي system في جسم الإنسان. أوكي، potent antigens to human immune system. بعد كده يجينا الكبسول. This is which is polysaccharide coating that surrounds the cell wall. Okay, it is this antiphagocytic virulence factors. Antiphagocytic يعني بيحمي الفنجاي from phagocytic cells. Okay. بيكون أكتر حاجة موجود في الكريبتوكوكس نيوفورمانس الكبسول ده، وthis capsule can be visualized with India ink stain. لما نصبغ الفنجاي for example كريبتوكوكس نيوفورمانس فنجاي بالإنديا إنك راح يكون عندنا جدا واضح الإيش الكبسول under the microscope. طيب السيل ممبرين هذا هو بيكون bilayered يعني طبقتين is the innermost layer around the fungal cytoplasm. بيحتوي على الستيرولز. اوكي، الستيرولز also بيوجد في السيل ممبرين of humans وفي البكتيريا. which bacteria mycoplasma bacteria بيكون فيها ستيرول. عشان زي كده أكتر ممكن ما نقدر نستخدم الانتي فانجل antibiotics. ليش؟ الفانجل antibiotics ليش؟ لأنه ليش ما نقدر we select this as as a target for fungal antibiotics لأنه ممكن يضر الhuman. Why? Because we have sterols. Okay. There is another sterol called ergosterol. Is essential sterol in fungi, while cholesterol is essential sterol in human. فبالتالي here we can use antifungal agents or medication to target this ergosterol because human cells don't have ergosterol. Okay, so the majority of antifungal agents work by disturbing ergosterol either by binding it to it بترتبط بيه and thereby punching holes in the fungal cell wall. Okay, تعمل له holes بالتالي they can distract the fungal cells. Example of these antifungal agents اللي بتشتغل على الإركوستيرول عندنا الإنفوتيرسين B and nystatin. Okay, or by interfering with ergosterol synthesis. في بعض الأدوية بتشتغل على الانترفيرنج تبع الإرجوستيرول synthesis زي الأزولز والإكينوكانتنز. طيب, what is the pathogenesis of fungal infection? بالمقارنة مع البكتيريا والفيرال والباراسيتيك disease, unfortunately, less is known about the pathogenic mechanisms and virulence factors involved in fungal infections. Still, we know little about the pathogenesis of fungal infections compared to bacteria, viral, and parasitic disease. But in general, most fungi are opportunistic, okay, and producing serious disease only in individual with impaired host defense system. يعني في immunocompromised patients أكتر حاجة بتعمل infections زي في AIDS patient, زي في الكانسر patients, يعني تكون immunity جدا ضعيفة ممكن يحصل إيش fungal infection and opportunistic infections. Only a few fungi are able to cause disease in previously healthy persons. يعني اللي بيكون عنده in healthy state كويس and good healthy state, they rarely can cause disease in these persons. But let's understand the pathogenesis here of fungal diseases. What is the portal of entry يعني mode of transmission? ممكن primary mycosis, post respiratory, inhaled spores, ممكن ingestion بواسطة الأكل, cutaneous and superficial, skin infection يعني contamination of skin, direct and indirect contact can lead to fungal infection. Also subcutaneous skin infection inoculated via بتدخل from skin trauma to the subcutaneous layer. طيب إيش virulence factors عند الفنجاي? They can produce toxin. But mainly in plant fungi. Okay, زي ما قلنا الكبسول في Cryptococcus neoformans is a very important virulent factor بيحميها. Antiphagocytic بيحميها من الphagocytic cells like macrophages. Also adhesions factors تعتبر as a virulence factor. Some of fungus had hydrolytic enzymes, so they are important as virulence factors and inflammatory stimulants. Okay, so these are the virulence factors of fungi. Morphological classification of fungi. زي ما قلت لكم هي بتعتبر dimorphic. Let's see more about the classical classification of morphological of fungi. some of them they are unicellular. اوكي يعني ايش unicellular؟ بتكون one cell بيكون دائرية spherical or ellipsoidal. الريبرودكشن تبعها بيكون biotic to form blastospores، تبرعم يعني، فحيكون شكلها زي كده، ده البرعم الصغير. okay. may form pseudohyphae if blastospores remain attached to each other. These are example of pseudohypha. morphology شكلها بيكون كده. متى بيصير ده الكلام؟ إذا البلاستوسبورز remain attached، يعني ما الدوتر cell doesn't detach from the mother cells، بيكون can form pseudohypha. okay. and spread easily through the host body. example of yeast cells عندنا Cryptococcus neoformans and Candida albicans، بس بتوجد بشكل ايش؟ yeast Cryptococcus neoformans and Candida. المولds also called filamentous or mycelia، كلها same as molds. formed of filaments called hyphae. okay. الهايفة بتتجمع مع بعضها بتعمل ايش؟ mycelium، تعمل mycelium. hyphae may be septate or accipitate، كلها هنشوف هنا example. عندنا السلايد اللي بعدها في example، فبتكون ايش؟ الهايفي هنا هايفي بتكون septate accipitate، يعني ما فيها septum. السبتم ايش هو؟ ده اللي هو زي الوول. اوكي. فهنا بتكون aseptum or accipitate hyphae، ما فيها اللي هي هذه الخطوط like walls. اما دي بتكون ايش؟ septate، بيكون فيها الخطوط هذه. طيب قلنا الهايفي interlace يعني بتتشابك مع بعض to form mycelium. this is mycelium. اوكي. يعني الهايفي، هذه الهايفي they are interlaced، يعني بيتشابكوا مع بعض، بالتالي هيكون لي mycelium like in this picture. طيب reproduce by what? by formation of conidia. عندنا two types of conidia, maybe unicellular called microconidia or multicellular called macroconidia. This is microconidia بتكون unicellular and this is macroconidia بتكون multicellular. okay. example of molds dermatophytes and aspergillus. okay. and here also pictures توضح لكم أكتر. اليست سيل بيكون شكلها دائري أو بيضاوي. okay. this is pseudo hyphae. okay. and this is a true hyphae. طيب في بعضها بيكون dimorphic fungi. dimorphic fungi يعني تغير شكلها بتغيير درجة الحرارة. okay. they can grow as yeast or molds. grow as molds when at 22 degrees 22 centigrade degrees تقدر تنمو as mold and also in soil. while they can grow as yeast لما ترتفع درجة الحرارة ل 37 درجة و in animal body. okay. يعني بيكون لها وجهين. okay. like what? like histoplasma and blastomyces. okay. وجهين according to the temperature and to the environmental settings. okay. طيب there are thousands species of fungi. عندنا آلاف الفنجاي. most of them are saprophytous. few species cause disease in man. Okay, fungi زي ما قلنا are eukaryotic organism and they can be classified clinically now. احنا قسمناها لكذا قسم. morphological هنشوف clinical كيف هنقسمها. clinical classification عندنا superficial mycosis, cutaneous mycosis, subcutaneous mycosis and systemic mycosis. لو قلنا عندنا thousands of them بيكون cannot cause disease in human or plants, لكن little of them may cause disease. Okay, so the superficial mycosis, these are fungal infections that are confined to stratum corneum. Okay, without tissue invasion. بتكون في الطبقة الخارجية من الإيش؟ من السكين في الستراتم كورنيوم. ما بتسبب أي tissue invasion. Okay, like what? Like Taenia versicolor اللي هو الملاسيلزيا فيرفر or Taenia nigara اللي هو الإكسوفيليا برينكاي. Okay, and this is an example of Taenia nigara. بتكون إيش؟ في حيكون discoloration في السكين. brown discoloration of من الهاند skin. Okay. طيب cutaneous mycosis هنا fungal infection will involve skin, nail or hair. هنا هتسبب tissue destruction and immunological reaction. For example dermatophytes. Okay. مين بتسبب cutaneous mycosis هي dermatophytes وبيختلف اسم المرض according to its site. For example هي Taenia corporis بيكون في البادي رينج وورم. اوكي. اذا كان في الجروين مين بيسببه؟ تينيا كرورياس نسميه جوك اتش. تينيا بيدياس تيجي في الفيت في القدم نسميه اثليتس فوت. تينيا كابيتس ان سكالب. تينيا انجيوم in the nail بيكون onychomycosis. onychomycosis. Okay, and this is example of ringworm. Okay, شكلها كده دائري as a ring. Subcutaneous mycosis. This infection confined to the subcutaneous tissue من اسمها but without dissemination to distant sites. Okay, only في subcutaneous tissue بتيجي and doesn't disseminate to other distant sites like mycetoma اللي هو المدورة فوت, الكرومومايكوسيس and sporotrichosis. Okay, while systemic mycosis من اسمها تكون حاجة systemic يعني أول حاجة بنلاقيها في البلمونري في اللنجز بعدين تنتشر ل any other organ. Okay, ومن اللي بيسببها dimorphic fungi such as Histoplasma capsulatum, Blastomyces dermatitis and Coccoides. Also we have here opportunistic mycosis. Okay, زي ما قلنا بيكون في immunocompromised patients. So the ristic mycosis are infections attributable to fungi that are normally found as human commensals or in the environment, such as Candida albicans, Aspergillus fumigatus, Pneumocystis carinii, Cryptococcus neoformans, and Cryptococcus gattii. These all organisms اللي ذكرتها ممكن cause infections in immune compromised patients like in cancer patients or AIDS patients. And then also there are two types of mycosis, تسبب allergies and mycotoxicosis. Okay, allergies to what? To fungal spores. The spores طبعا ممكن تسبب allergies to human, particularly of Aspergillus. Mainly type 1 بتسبب type 1 hypersensitivity reactions or atopy, manifesting as bronchial asthma, hay fever, urticaria. Also, they can cause a mycotoxicosis, okay, which due to consumption of food containing fungal toxins. Okay, بياكلوا food بيحتوي على fungal toxins. Okay, for example, after eating mushrooms ممكن يسبب hepatotoxins في الليفر. Another one like ergotism is caused by mold called Clavispus purpura, which infect grains, grains and produce alkaloids اللي هو الأرجوتامين and lysergic acid diethylamide اللي هو LSD بيحتوي. Alright. Other toxins ingested with spoiled grains and peanuts اللي هو الافلاتوxins بيوجد في peanuts وال spoiled grains which are metabolized in the liver. Aflatoxin are coumarin derivatives produced by Aspergillus flavus. They are hepatotoxic, causing hepatic carcinoma in man. So very important this information. Okay, يعني واحد ما بيستهين بالفنجس because they can also cause cancers in men. Okay, hepatic carcinoma in men. But how can diagnose fungal infections by using laboratory techniques? طبعا أول حاجة لازم ناخد عينة اللي هي السبيسمن according to the site of infection. إذا كان في skin هناخد skin. إذا كان skin infection هناخد skin specimen. كان nails infection هناخد nails specimen and so on. Okay. Also maybe pus, blood, spinal fluids, sputum, tissue, biopsies. Okay. And then we will see it under the microscope using what? Using potassium, 10% of potassium hydroxide. Why we use potassium hydroxide specifically? Because it dissolves keratin موجودة في السكين والnails but doesn't affect fungi. Okay, ما بيأثر على الفنجاي. Branching hyphae are detected among epithelial cells. ممكن نشوف برضو الhyphae under the microscope. Fungal stains such as lactophenol ممكن برضو نستخدم lactophenol cotton blue stain. Calcofluor white stain could also be used. Okay, and this is one of the fungi called Aspergillus. Under the microscope, we stained them by lactophenol blue stain. Okay. Okay, also we can culture the fungi using saprodextrose agar. تتكون من ايش؟ دي المادة أو الميديا بتتكون من 4% of glucose, 1% peptone, 2% agar, and the pH بيكون 5.5. Why we use this type of pH media? Because it inhibits bacterial growth while allowing fungal colonies to form. عشان we prevent the contamination with other microorganisms like bacteria. Okay, so it doesn't affect the result. Also, we can use chloramphenicol plus cyclohexamide to further inhibit bacterial formation. are incubated separately. ليش؟ عشان ما احنا قلنا في حاجة اسمها dimorphic fungi. ف one at room temperature 25 degrees and the other at body temperature at 37 degrees. Most yeast grow at 37 degrees. أكثرهم بينمو at this temperature. Okay. Molds grow at room temperature for up to 4 weeks. طيب how to identify isolated fungus on culture media. For molds, identification is done by microscopic examination. يعني we don't need a microscope. We can use our eye to examine the fungus. بنشوف الكولوني وبنشوف color on surface and reverse. Like here, for example, see this. We don't need any microscope. Okay. So how to identify the isolated fungus on culture? For molds, احنا قلنا المولدز اللي هي بيكون only yeast. Sorry, only hyphae بيكون شكلها hyphae. فالidentification هيكون microscopic by using your eyes. We don't need the microscope because you will see the colony morphology فهيكون واضحة. ونشوف الcolor. Okay. For example here الترايكوفوتون rubra الcolor تبعها على on the surface بيكون لونها أبيض قطني. لكن on the reverse or the back of the plate لو قلبنا البليت هيكون لونها أحمر زي ما انتم شايفين. Also we can use microscope to study the morphology of conidia. عشان أشوف الكونيديا for example here I need the microscope. I can't see it by naked eye. طيب for the yeast اليست اللي قلنا الدائرية برضو بيكون by microscope by your eyes على البليت على SDA plate اللي بتنمو عليه بيكون شكلها زي كده دائري creamy colored. اوكي بيكون لها رائحة معينة معروفة نسميها yeasty odor. Or microscope using microscope وبيكون شكلها like this oval دائرية أو بيضاوية الشكل gram positive يعني لونها أزرق or purple in color. For example here Candida albicans. Also we can use some biochemical reactions to identify them. For example using germ tube test, chlamydospore formation and sugar fermentation. Furthermore, we can use serological tests and immunofluorescent techniques. Okay, we use them to identify fungi from clinical isolates. Okay, clinical isolates اللي بنحصل عليها from the hospital, from the patients from in hospital. Serological test to detect antibodies, okay, against fungi. Detection of rising titer is more indicative. Here for example to diagnose histoplasma هو المرض اللي بتسبب بنسميه histoplasmosis. We detect antibodies against two antigens. إيش هي two antigens؟ اللي هي histoplasmin antigen وال inactivated whole yeast phase cells. Okay. التست اللي بنستخدمها complement fixation test and immunodiffusion test. So these are the two serological tests. Antifungal medication, what we use to treat fungal infections. طبعا the number of infections زادوا dramatically with increased patients who are immunocompromised from AIDS, from taking chemotherapeutic drugs, patients from who undergone organ transplant and they use organ transplant immunosuppressive drugs. So for this reason, you will frequently use antifungal medication. Okay, زي ما قلت لكم we have thousands and hundreds species of fungus, but most of them can't cause disease. Little of them cause disease, but in immunosuppressive patients. Okay. Ergosterol احنا قلنا this is the most common sterol in fungus and it is an important part of the cell membrane of fungi and fortunately is not found in the cell membrane of humans. So we can use safely this drug in humans لأنه الهيومنز بيكون عندهم cholesterol. So most of antifungal agents bind more okay, avidly to ergosterol than to cholesterol. بترتبط أكتر بالإيش؟ بالergosterol. More selectively damaging the fungal cells than human cells, so it is safely to use it. By binding to وسطة binding or inhibiting ergosterol synthesis, they increase the permeability of the cell membranes, so causing cell lysis and death in fungus. Antifungal agents can be divided into 6 major groups based on their mechanism of action. يعني نقسمها to 6 major groups according to their mechanism of action. على حسب mechanism of action. For example, here prolines بيحتوي طبعا على الأنفوتيرسين B and it is very classical. هو أقدم دواء عندنا في antifungal treatment. So it forms what mechanism of action تبعه forms a complex with ergosterol. يعني بيشتغل على ergosterols بيعمل disruption of fungal plasma membrane which lead to leakage of cytoplasmic contents and then fungal cell death. Okay, and it covers the majority of medically important fungi. بيأثر على the majority of fungi. Okay. Second group called antimetabolites. Okay, وعندنا only one agent in this group اسمه flowcytosine. Flowcytosine. The third group called azoles, and their mechanism of action is to inhibit the fungal cytochrome P450 3A dependent C14 demethylase, which converts lanosterol to ergosterol. So again, بيأثر على الergosterol يعني, leading to depletion of the ergosterol in the fungal cell membrane. So this group includes clotrimazole, fluconazole, itraconazole, alvoriconazole, البوسيconazole, and آخرها بيكون azole. Okay, this class is used extensively as it has less side effects to compare to the other class. لأن أكتر حاجة بنستخدم الأزول لأنه بيحتوي على less side effects compared to the other class of antifungal treatments. The fourth group is called glucan synthesis inhibitors, معروفة بإيكينوكاندنز. These newer agents, which include caspofungin, تحتوي هذه المجموعة على caspofungin, mecanofungin, anidilofungin. So they inhibit the cell wall synthesis. يمنع cell wall synthesis بواسطة inhibiting enzyme 1,3-D glucan synthase. بيأثر على الجلوكان من اسمها. Okay. فبالتالي هيأثر على السيل وول synthesis. The other group is called allylamines. So this class consists primarily of topical antifungal agents, okay, with exception of terbinafine, which is topical and oral. Okay. These agents inhibit also the ergosterol synthesis بواسطة inhibition of squalene oxidase, which is important to ergosterol synthesis. So if we inhibit the squalene oxidase, we will inhibit the ergosterol synthesis. Others group, for example, grazoflovin, which is a new antifungal treatment, the cell wall division by disruption of fungal cell mitotic spindles. Okay, so these are additional resources. You have to go through it and read it and study it carefully. And thank you for your attention.